Today we are fortunate to have with us Professor Shubhra Chakraborty, who is a very accomplished scientist. She is the director of the National Institute of Plant Genome Research in New Delhi, Aruna Asaf Ali Mark, which I happen to know very well. She is the director since 2020. In her illustrious career, she has received many awards and honors. It's a pleasure and a privilege to introduce her. She works on nutritional genomics and stress genomics. Her research, unlike a mathematician like me, her research is extremely practical and has the noble aim of improving the nutritional quality of important food crops. So it relates to our day-to-day life, affects our day-to-day life in a better way. As I mentioned, she has received many awards and honors throughout her career. I just mentioned that she's a fellow of the Indian Academy of Sciences as well as of the Indian National Science Academy, and she's a recipient of the J.C. Bose Fellowship. With that, Professor Chakraborty, we are eager to hear from you about plant diversity.
Thank you for your very kind introduction, Professor Bhattacharya. And at the outset, I would like to thank the Academy, the National Council of Science Museum, and also the Academic Trust, and particularly Professor Partha Majumdar, the President of the Indian Academy of Science, to give me an opportunity to actually come and speak about the plant diversity, which is very close to my heart. And I think we all live on this earth, and plants are those organisms which actually make us to live on this planet Earth. So I'm really grateful, and I hope that I would be able to do proper justice to this. And while actually preparing this, after many years that I have finished my botany post-graduation, I have now learned many things because of this talk today during the preparation. So this is a talk. Before this, we have also heard Dr. Rainey Borges, who spoke about the planetary evolution, as well as after that, we also heard another talk, very nice talk on animal diversity. Today, I'm going to speak to you on plant diversity. So if we start talking about plant diversity, before that, I think we need to understand a little bit of the concept of biological diversity. And you know, the planet Earth that we all see and inhabit today was discovered around 4.54 billion years ago. While I'm sure all of you know that life on Earth was found 3.5 billion years ago. And as we know today that the ecosystem on this planet comprised of prokaryotes and eukaryotes, and those eukaryotes includes the plant and animals, it was a great philosopher of all times that we know, Aristotle, way back in the year 384 to 322 BC, classified actually animals according to their living environment as either aquatic or terrestrial. And he further then divided into two groups, blood and bloodless, which was basically today's animal and plant. Contemporary to this, if you see the history, in the year 371 to 287 BC, another great philosopher, and whom we all know as father of botany, Theophrastus, created and maintained a first garden on the grounds of Aristotle Lyceum. And you could see in this slide at the bottom that Lyceum. So what happened? Actually, Theophrastus wrote two books at that time on major botanical works that he did. And one was De Historia Plantarum, and the other was, Causus Plantarum. And this first work actually described the anatomy of plants and classified them into trees, shrubs, herbaceous perennials, and herbs. Whereas the second work discussed their propagation, growth, and served in part as a practical guide to the today's farmers and gardeners. Thereafter, what happened? Long years after, from the 3rd century to the 18th century, in 1735, it was Carolus Linnaeus, the father of taxonomy then, published his most innovative work, which we know as the System of Nature, Systema Naturae. That provided actually the framework of the two kingdoms that we know today as Kingdom Plantae and Kingdom Animalia. And then, these were, he did it based on the basis of nutrition and mobility of those kingdoms. Organisms belong to those kingdoms. And however, there were certain drawbacks that was later highlighted by the later botanists, and those were that there was no clear distinction between the eukaryotes and the prokaryotes in his classification. And also, he kept the fungi within the photosynthetic plants at those days. And this classification was then too broad and demanded a little more specification. And then what happened next, what we found, it was the Darwin who actually came, and as you all know, that he did the journey towards decoding the evolution, and a very famous Galapagos Island that he went days after days and months after months to understand all the organism inhabiting at that time in that Galapagos Island. And that was a first journey towards decoding the evolution, we would think. And in that case, as said by Frans Lanting, actually a Dutch photographer, that biodiversity starts in the distant past and it points towards the future. And this is exactly what we still see, whatever Darwin did. And the Galapagos Island therefore provide a window on time. And in a geological sense, although the island are still young, but yet they appear very ancient if we go through the history and read about the botany. Now, what happened, that we know that Darwin actually wrote his very famous book, Origin of Species, in 1859 and gave his theories of evolution. However, one of the most prominent example that he stated was that of Galapagos finches. And their big shape, which is known till today by all of us. But then, however, most of us possibly do not know that we have Darwin finches also, and which is nothing but Scalacia. And the plants, those you are seeing the bottom of this panel, those are Scalacia affinis, and they are known as Darwin finches. And if you could see that even it is the same plant, it is the same taxa, it is the same, genus and species. However, just see at the right hand bottom corner that the same plant can have so much of variation only in the leaf shape. And the variation in the leaf form within the genus Scalacia actually wondered Darwin and also wonder us even today. And this is the famous Darwin finches of the plant world. Now, thereafter, what happened is that Darwin tried to understand what he is seeing. And while he was going through all these species, he also saw what he saw was that is known as today abominable mystery. And what Darwin really meant about that, he coined this phrase in 1879, and in a letter what he was writing in those days to his very close friend and also another botanist and explorer, Dr. Joseph Hooker, he wrote that, I quote, "The rapid development as far as we can judge of all the higher plants within recent geological times is an abominable mystery." Why did he say so? If you see at the right hand side, the whole earth came through the evolution from the Carboniferous era to the Jura, Trias, Cretaceous, Eocene, Miocene, and now we are in the Quaternary. But then what he observed, to his surprise, he saw that there is a very long gap, which nowadays we do not believe, but at that time Darwin believed that. And then he wrote to his friend that why isn't there a gradual evolution of the angiosperms? Why can't we see intermediate forms between the gymnosperms, things like conifers and the flowering plants? And why, when they appear, are they already so diverse? That is where he was wondering in those days. And then what happened thereafter is that what we find, then the classification and the kingdom concept started evolving, and that was Herbert Copeland in 1902. Between that period of 1902 1968, he came up with the next concept from the two kingdom concept to the four kingdom concept. And I would urge all the students who are hearing today, please go back, although this is a very, very old manuscript published in Science in 1969 by Whitaker, whom I will be taking in my talk a little later. It's a very, very nice paper in the Science magazine, which talked about this whole evolution of two kingdom to four kingdom to the five kingdom concept. So what Copeland did, Copeland actually proposed this four kingdom classification or the concept, and which included Monera, Protectista, Plantae, and Animalia. And thereafter also, what we find in this, even despite he included the plants and fungi as a different kingdom, but then there was some more, refinement needed to be done, because the symbiotic organisms, the lichens, were given no kingdom at that time, and also there was some ambiguity in the angiosperm and gymnosperms classification at the time also. So thereafter, what happened, it was then the Whitaker basically who came up and who gave the five kingdom concept, which till today we follow, and that is, as you all know, that Plantae, fungi, Animalia, Protestia, and Monera. And here the plant and fungi are very different, only because both of them have, the very unique cell organ, that is the cell wall, which is not present in other three kingdom, but still the fungi cannot photosynthesis because they do not have the chlorophyll, and therefore we needed another different kingdom classification, and this is what Whitaker actually contributed to it. And then if we then go on and try to now see at the geological time scale how all these plants evolved in this planet Earth, and how the diversity, fantastic diversity that came in the plant species, as you could see here, we started from the Paleozoic we moved to the Mesozoic, we came to the Cenozoic era, and as you could see, starting from Ordovician till today to the Quaternary, millions and millions of years it took through the whole great evolution. And evolution is always that is what gives us the diversity, because of so many criteria. One of that major thing is the mutation, and then that's how we actually got all these plants today what we see in the planet Earth. I don't have much of the time, but if you could see from the right hand side, the diagram, you could see small photos of all those, starting from liverwort to the moss to the cycad to the kind of the conifers to the gymnosperm and the angiosperm, how over the million years they have evolved, and they have come with so much diversification, which I'm going to tell while I go on. Now, what happened when we now see that seed plant originated? There are diverse seed plant that came in the phylogeny, and in those seed plants, what we see actually, Linkis showed that in his very new paper, not new, I would say almost a 10 years back paper in New Phytologist, very nicely, he has shown that how the seed plants evolved and how the diversification happened over the years, starting from the Devonian. And we see now the concept of gymnosperm and angiosperm, how these came into our classification or the kingdom classification. And the angiosperm that we all know have also two clade, one is monocot and another is a eudicot. And then evolutionary history and relationship among this group of organism then came, and there was a sudden surge in the vascular plant, and that we have discussed little earlier now. And in that case, it became very interesting to understand not only the evolution, but along with the evolution, how plants have diversified across the globe, and that becomes very, very interesting aspect. If we try to trace back towards the history, how these histories were discovered. You know, we are talking about BC, that is before Christ, and today we are in a 21st century. How do we trace back what was there at those times? It is actually the paleobotany, and that means a fossil history that takes us back to so many million and billion years ago. So you know, I was reading, and to my surprise, I found out that world's oldest plant fossil that has been actually discovered in India, and that is a crown groove rhodophyte. We all know that plants and evolved from algae, but it was this rhodophyte which discovered in India is now told that it is the world's oldest plant fossil, as you could see at the upper panel left hand side. And the right hand side, if you see, it is actually a leap of faith, which is that how then the green algae comes and gives today's first land plant, and that is Rhynia, that came as a first land plant in the evolution. And if you see, there is so much of deviation, so much of variation you could see in the rhodophyte as a fossil, and the leaf structure, if you see here, pinnate leaves, and then right hand side, if you see the Rhynia now, you have a very hard stem, the leaf structure is not there. So it's so much of a diversification even at the organ level that we find. If you now see, there are many other fossils. I'm just putting few of the examples which are very famous examples. So you could see the lepidendron, you could see the seed fern, you also see the early Cretaceous epoch, and that is Artifactus. And this is where the paleobotany tells us that what was there before we even have born. Now, if we try to see today, where do we stand and what's the global diversity that we have? It is said that we have somewhere around 4 lakh total plant species on this globe Earth. And if you see that the left hand side categorize them depending upon the availability and the density of those diverse plant species over the globe. As you see, it moves from the green to the red. So the green is the areas where you do not have that great diversity of the plant species or the density of the plant species. However, as you go towards the color scale of the yellow to the orange to the red specify the most dense and most diverse plant species in those regions. And if you could see, India falls somewhere in the orange range. And in that case, you know, the most diverse species that we get is the South America, and also somewhere in Africa, and also in Australia. But India just comes after that. And if we try to see, I'm not going to read out the numbers, but we could actually divide them in the vascular plant, bryophytes, flowering plants, seed plants, so on and so forth. Now, if you try to move further, what I said, that the ecologically and biogeographically, you see a very different pattern of plant diversity across the globe. We can actually divide them as their taxonomic or as they are found. Vascular plants, endemic vascular plants. Endemic means that it is there in one region. You also get some rare land plants which are not there in many places. However, it is basically 3 of the continents that actually are the, I would say, the hotspot of getting all these vascular plant, endemic vascular plants and rare land plants. Then those are nothing but neotropics, Africa and Asia. Within Asia, India is one part that we all know. As I would be going through in my next few slides, I'll tell you that. And now, recently, globally, the biodiversity hotspots have been categorized. And in total, there are 32 biodiversity hotspots, as you could see throughout the globe. Now, out of all these things, what we find in India, when we talk about, do we have any kind of biodiversity hotspot? Yes, indeed. India have 4 out of 34 biodiversity hotspots today. And if you try to see, if I take you back to the recent publication from the Indian Agricultural, Indian Agricultural Research, and in that case, you know that India has the 2.3% of the total geographical area over this globe, right? And we have all the total world's 4.2% water resource. We have a 1% forest cover, pasture is 0.5%, and we all live on a 9% arable land in our country, that is India. If you see, out of all these, the 55% of that 9% arable land belongs to agriculture, whereas the rest of the things, as you see at the left pie at below, goes for the wetland, water bodies, snow and glaciers, grazing field of the animals, forest cover 22%, just after the second major portion after the agriculture land. And if you try to see the ENVISTAT data, which came up from time to time, it comes up, and the 2018 data shows that we actually are not having that much of region in 2015 to 2016, which we had possibly as an open forest and moderately dense forest even in 10 years back. So somewhere we are also losing diversity because we are losing our land space. So where those new species will grow. So if then you try to categorize the biogeographically the zones within all these 4 hotspots, and these zones have been categorized at the level of the province, and we have 10 such biogeographic zones. I'm not going to read out, but it starts from Himalaya, goes down to the Kanyakumari and eastern region to the western coast, that is the Western Ghat. So we have 10 such biogeographic zone in India. Now, how these all new plants, endemic plants, are there in these 4 hotspots. If we try to take account of the plant species that we have in India, the left-hand side map shows you we have more than 7,000 algae, around 1,200 pteridophytes, 2,500 bryophytes. We also have 18,000 angiosperms, and we have 74 gymnosperms, and somewhere around 2,400 lichens. And the area, again, as you could see, depending upon how diverse, how dense they are, it goes from the orange to the very high densely populated, as you could see in the northeast region, and that is a very densely populated plant species that we get in the Indo-Burman region. And that Indo-Burman region is one of the hotspots, in addition to the Himalaya, in addition to Western Ghat and the Sunderland. But it's very, very interesting to note that all these 4 regions have very different type of plants. Why? Because as we all know, the environment differs a lot. In the east, in the north, you see the glacier, you see very cold weather. As you come along and go downward to the Western Ghat, it's more humid and the temperature is much higher. As you then move to the Sunderland, it's another atmosphere. You move to the northeast of the India, and in that case, you see again very high rainfall, and therefore it's again a humid zone with high temperature. And therefore, you know, the ecological niche of any organism actually makes them what they are, and where they grow, they come into such kind of clades and such kind of consortium that they grow in those areas. So therefore, what we observe, beautiful flowers in the Himalayan hills, we then see kind of, you know, in Western Ghat, we see some other kind of vegetation. As we move to the eastern and to the Sunderland, it's a very, very different vegetation. I would like to draw your attention on one of those, that is, you know, Rafflesia amaldifi. So these are so much so diversity our own country has, that still we have a lot of treasure trove that we still need to explore. Now, all these 4 biodiversity hotspots have been also categorized into megacenter and microcenter of many endemic plants in Indian subcontinent. And we, as I just shown you, that there are 3 such megacenter: Eastern Himalaya, Western Ghat, and Western Himalaya. However, we have many microcenters starting from Andaman Nicobar Island, and it travels to this continent, as I say, that encompass Nepal, Bhutan, and many other countries. However, right-hand side, if you see, we have such nice glacier in the Darang Darun, we have alpine vegetation, we have Quercus forest, we also have deodar forest and pinus forest. And as you go on to the tropical to the temperate zone, you find all these diversity in the plant kingdom, and mostly they belong to conifer and the gymnosperm. So if we now then try to see that what are the plant groups that the latest botanical survey has come up last year, and that report tells us that we have in India somewhere around 4 zone. Northeast region harbors 19%, Western Ghat 35%, Eastern Himalaya 22%, Western Himalaya 24% of all these species. However, in last 1 year, that is in 2020, 267 new plant species have been discovered in India, of which 119 are angiosperms, 3 pteridophytes, 5 bryophytes, 44 lichens, 57 fungi. I should not take fungi in plant. However, this data tells that 21 algae, and also there are 18 microbes. So therefore, the seed plants came as 42%. That is the maximum majority of the plant within the plant kingdom, and that we find. So as I was telling you, the ancestral eukaryote, which actually gave in the evolution all different forms of plant species, are nothing but the green alga. And this green alga and the brown alga actually gave rise to, in one direction, the non-vascular plants, which are mosses, liverworts, hornworts, and the multicellularity and the tissue differentiation started at that time. Also, what we find, these non-vascular plants have the ability to regulate their water loss. You know, there are some Selaginella, if you have heard of. If you actually take Selaginella, which is a non-vascular plant, and put them in a desiccated condition, then all leaves will be crumpled, and they would become a very small plant. The moment you water that plant, it then blooms, and you see such nice ramification of the plant leaves for Selaginella. And that is the nature's creation, that it gives the power to those plants because they have to survive such conditions, and they can regulate the water loss. And then beyond that, we also have the first vascular plant that I've shown you in the earlier slide, Rhynia. And then from there, the Rhynia gave rise to the first seed plant. And then in the other case, what we find from the Rhynia, that the vascular plant evolved, and we have the non-seed vascular plants and seed setting vascular plants. Non-seeds are ferns, seed settings are, as you can see, conifers and the flowering plants. And in the flowering plant, we have that they have now evolved the capacity of protecting their seeds through the flowers and the seed for their next progeny. And they also have the secondary growth that is related to the tissue differentiation, tissue diversity, I would say, which is cambium, and which is strong xylem and phloem. When we see to the vascular tissue of the conifers, they still don't have a kind of they have still gametophyte protection mechanism and the seed formation mechanism. Now, with that, when we move to this, what this slides is only for the, the students who are with us. And I am told that there are school students and must be having in your school book, this all the things I have just put it into a table format, that how this algae to the moss, to the fern, to the conifer and the flowering plant, when they evolved, how the anatomy changed, how the morphology changed, how their function changes. For example, algae has the water for fertilization. Mosses have rhizoids. We don't see rhizoids in the fern many a times. Sometimes we see them, but what we don't see in the fern, that is the seed and the cones and the fruits, which then comes in the conifer. And if we go to the flowering plant, cones are then vanished, and it is only the fruit and the seed. And that's the diversity. That's the huge diversity that we see in the plant kingdom. Now, if you see that, it's a beautiful quote that says that the diversity where is the beauty lie, and there is the strength that is also lies in the diversity. And plant world is such thing which gives this diversity and gives the strength for everything. You see a giant sequoia. That is the world's largest plant that you see in California, whereas this duckweed, which is the world's smallest flowering plant. So you see, one is feet and feet tall. Another is maybe in centimeter. So in a scale of the growth, they are so differ. They differ so much. You see a ginkgo biloba, which is a living fossil, whereas we see maple seeds. We've never seen this kind of seed, you know, which has a winged fruit, and we see, you know, legume, you see kind of total fruit, but sometimes this possibly came from fruit as such, but you know, the seed is inside. But here you see the maple seed is just like a winged fruit. Now ephedra, which is a source of ephedrine drug, and this flowers gives the drug that we actually use. Selaginella, I have already talked to you, and therefore it is known as a resurrection plant today. Sago palm is another wonder of the world, and these are beautiful diversity that we see not only in color, but in shape, in size, in their function. Now, I would take you through little bit, few slides now, that what could be the facets of this diversity in the plant kingdom. There are diversity in the plant kingdom. When we talk about the organ diversity, you know, there are vegetative organs, there are reproductive organs. Even within the vegetative organ, we have the root, we have the leaf, we have the, you know, there is so much difference in the root. When we talk about the root of the angiosperm, of the gymnosperm, of the monocots, of the dicots, you find there is a primary root, there can be a fibrous root, adventitious root, aerial root. You have also a taproot, and taproot getting converted to a food, and that is carrot, you know. And in that case, when we come to the leaf, you again see a simple leaf, a lobed leaf, a compound leaf, a palmate leaf. So there is so much of diversification. We move on to the stem. You see underground stems, aerial stems, subaerial stems, and all the underground stems are again something where one organ goes and becomes a storage organ, and therefore a food we take, very example, ginger, potato, every day we take, and they are all modified stem, and they are grow all underground. When we come at the aerial, where you have the sunlight, you actually see Dioscorea bulbil. You see the thorn. You see the stem is going as a thorn in Opuntia. It is just a mechanism by which they can actually cope up with the water loss, because they grow all in the desert zone. And then if you come to the sub-aerial, you see a runner, you see a sucker, you see a stolon, and so on and so forth. We move on to the plant reproductive organ diversity. As you know, reproductive organs are two. One is flower and one is the fruit. And in that case, you see such nice, beautiful flowers. You see not a single flower look alike to the next flower that you see, not even in shape, not even in color, not even in their, you know, the holes of the petals or the carpels or androecium or the gynoecium. They all are diverse. When we move to the fruit, you see starting from the mango to the, you know, the anar, to the banana, to cucumber, to ananas, wherever you go, it's such different and such different in taste. I would link this thing to the, you know, chemical diversity later on, and let's go and see that how then, when we move from the organ diversity, we come to the plant life, yet different diversity we observe. Basis of habitat, as I talked to you, hydrophytes, halophytes, xerophytes, hydrophytes, mesophytes, epiphytes, parasitic plants. When we move to their basis of the habit, you see the tree, herb, shrub, climber, trailer, palms, creepers, so many. And then as you move again to the diversity based on the lifespan, you find annual plants, you find biennial plants, you find perennial plants, ephemeral plants. When you move to their nutrition, you see then insectivorous plant, you see then parasitic plant, you see then saprophytes. When you go to the basis of their size, you find largest to the smallest. That I have already talked to you. Now, when you actually come and try to see, is there only morphological diversity that we observe in the plant kingdom, or there are some other kind of diversity? You know, it's very interesting to study biochemistry. And when you study biochemistry, what you find, the diversification of the plant kingdom never only happened because of the morphophysiological diversity or anatomical diversity, but it also primarily because of the primary metabolism diversity that has happened. And this is the gift to the chemical diversity that we plants have been gifted with. And with all this chemical diversity, as you could see, the blue letters here actually shows you the primary metabolic pathways, whereas the green letters are those downstream specialized metabolites which comes from the primary metabolic pathway. As you go and see the blue boxes that I have marked as IPMS3 and so on and so forth, those are nothing but alternative routes of enzymes or enzymes to synthesize primary metabolites, and then you have green arrows. Those are the pathways derived from specialized metabolites. When you dotted lines, they are feedback inhibition. Now, while this diversity, chemical diversity, which is huge, we know genes, we know transcripts, we know proteins, we know metabolites, but let me tell you, metabolites are the most diverse class of 4 of the biological molecules that we have, and these metabolites actually are very much responsible for giving us all the phenotypic diversity that I have shown you in the earlier slides. And just to give you an example, photosynthesis we all know, and however, plant has 2 different kind of photosynthesis. Many of you would be knowing C3 photosynthesis and C4 photosynthesis, example rice and maize. And why this happened? Because they grow in different atmosphere. They need to reserve their food properly. And therefore, nature went ahead and changed the metabolic pathways. All the Rubisco is the same, but the biochemical activity of the Rubisco of the Rubisco is so different that it gives 2 different mode of a diverse physiological and biochemical pathway, even for the well-conserved phenomena, and that is nothing but photosynthesis. When we then try to see who were the Indian stalwarts in the field of plant diversity that actually have worked relentlessly and given us the concept of many of these things, from starting from the algae to the angiosperms. It started with the O.P. Iyengar. He actually worked many years on the algae, and then what we find that there are many other people, starting from Birbal Sahni, who actually was a paleobotanist. In his name, we also have an institute in Lucknow now. And also, thereafter we had Janki Ammal, who is actually the Indian botanist who worked on not only to the plant breeding and cytogenetics that we know, but if you go to the literature, you would find that she was a phytogeographer, because she moved from the India to UK to US, worked with C.D. Darlington, and then came up with her famous book of Chromosome Atlas of Cultivated Plants. Why I'm taking this here? These chromosomes are have the hereditary units, right? We know that, and these chromosomes therefore are the primary point of origin of diversity, and that is what Janki Ammal those days actually have told us. One great person that I could still remember, my teacher, Professor Archana Sharma, who also contributed on understanding of chromosome and chromosome-based taxonomic classification, which I have also been taught by her, and I was a lucky student of her. So if we move, then we also have Devabrata Chatterjee, who was a botanist from India and whose primary scholarly focus was on the endemic flora of India. We also have Gopinath Panigrahi, who was another botanist and plant taxonomist, majorly contributed in the establishment of tragus as a new species in India that we know. And lastly, if I try to connect the plant with the chemical diversity, I cannot forget one great lady of Indian origin, and that is Professor Ashima Chatterjee. And she actually shown us so much diversity in the organic chemistry, which she always worked in the plant to find out organic chemistry, how the pathways happened, how the molecule came, and it's a great discovery, many discovery that she had done for, and that led to the organic chemistry and the phytomedicine of today. So coming to those were the people who actually gave us all the concept. Now, still we have in our newer generation people who are still, enthused and intrigued by the diversity of the plant. And these are few of such discovery that has come only in last year. So one work came from ISAR, and then some other work came from some other researchers, from Ritesh Chaudhary's work, from some others' work, and where they actually found out many diverse species from our country. Not only that, as you could see in the Times of India coverage, new plant species discovered in Antarctica named after India, because it is an Indian who discovered that plant going to the Antarctica. So it's a huge diversity, untapped even today, as you could see, and I'm sure you're convinced from this slide. These are few such examples which came only last year, and you could see there are algae at Himalayan. It is Fortia, it is Luticola. When we come to the fern, there were 2 ferns were discovered last year. We also had 4 more bryophytes discovered, many other seed plants discovered only last year, which I took from the Botanical Survey's latest plant discovery report. And in that, as we go on and try to see, is it that only the planet Earth where the plant is growing? Yes, indeed, no. Not today. It was there yesterday, but not for tomorrow. Tomorrow, people are thinking that whether we will be having some other planet to inhabit it may not be today, but in the near future, maybe down the line, 10 years, 50 years, 20 years. So people are trying to see if that happens, how do we have more diverse plants? What are the plants which can cope up on such kind of atmospheric condition? And NASA has started this kind of a work. And if you see, there is a Kennedy Space Center. Many of you might be knowing. Those students who do not know, please go back and see. It's very fascinating work that is being carried out by not only NASA, but in collaboration with participating different space agencies like Roscosmos, ZACA, ESA, and CSA. 5 agencies together, they are into this work, and the study will add to the NASA's knowledge of growing food crops for long duration space missions. So if you have to send a spacecraft, if those people who are going there, how do we provide them with the food that they can sustain? Tomorrow, if we have another planet coming up, we never know what's going to come up, and we have to go and inhabit it. Can we have preparation for those in future? And in that case, it is basically the vegetable production, and nowadays we call it veggie, and those veggie gardens, and they actually try to take pepper as one of those. And it has been shown that pepper has several key nutrients and an excellent source of vitamin C. So can we produce vitamin C from the pepper? And that is again a chemical diversity that we have seen in the earlier slides. So now, little bit, if I would like you at the end of my talk, would like you to connect that this enormous and fantastic and fabulous plant diversity that we have seen, can that be linked to the agrodiversity? Yes, indeed so. If you actually see that what's happening today in the agrodiversity, since 1900, some 75% of the plant genetic diversity has already been lost. And farmers worldwide, it's not only in India. And because farmers have left their local spaces, and then local varieties are not there. Land races we do not get nowadays, and which are actually the variation, which are the genetic variability we are losing. Second is, today 75% of the world's food is generated from only 12 plants. I have shown you we have 4 million plants, but what we use today's food comes only from 12 plants and 5 animal species. If I take you to more, then of the 4% of the 250, 100,000, 300,000 known edible plant species, only 150 to 200 are used by humans today. So you see, we are not utilizing many of the species for the human benefit. Now, only 3 of the crop, and that is our staple food today, not only in India, but globally, of which rice is feeding 2/3 of the global population. That is rice, maize, and wheat, and that contribute nearly 60% of calorie that we consume and the protein obtained by humans from plants. And then if you see that because we are losing wild relatives, we are losing wild edible species, we are having domestication and semi-domestication of the species, we are losing, and therefore there is a great loss that's actually connected with the plant diversity and the agrodiversity. There are many reasons for that, and some of those are written here. I'm not going to read out because of the time constraint, but let me tell you how this crop diversity can play a role as a treasure trove for our food security, that we keep on telling food and nutritional security, so important for everybody to live, not only the human, but the animals, and also for the plant, for themselves, because they also have to live. Now, in that case, this slide very nicely shows that there are millets, there are legumes, there are cereals, there are, the crops which are coming from the tuber crops, which actually gives, and the fruit crops and vegetables, which are the treasure trove for the crops as for our food security. As I told you, that the chemical diversity of the plant has also been exploited and utilized for the application in pharmaceuticals and nutraceuticals. If you see, it started from 1800 when morphine came from poppy, ephedrine from ephedra. We go on to 1899 to the 1900s, and you see to the 2000, we have so many plant-derived, phytomedicines, Raulfia, we have vincristine, we have artemisinin, which got a Nobel Prize just a few years back from a Chinese lady. And then you have the lovastatin, you also have nicotine, curcumin, you also then move. But what happened? Up to 2009, all those things have been taken not only in our country with FDA and many other places. However, thereafter, there are only few such have been now on trial, and last few years we haven't come up with more diverse phytomedicine. So this is an area which is totally a barren area in last 5 to 6 years, and possibly we need to look into it more carefully, so that can we discover many more new drugs from the phytosource. And I'm sure if some of you who are younger generation can take interest, and trust me, they're so interesting. And if you go to understand their biochemistry, it's such an interesting thing with the genetic diversity that they have and how the genes and proteins regulate these pathways. Very interesting, fundamental research that one can do, and that can give out to the application or the translation for the benefit and the safer health for this earth. Now, little bit, if I try to connect you with the plant diversity as a very valuable management tool for the farmers. So what does it mean? It means there are 6 graphs that you are seeing. All these graphs have been actually shown here as a plant diversity species. So you have this kind of diversification, how they are linked to the metabolizable energy yield, how they are connected to the utilizable crude protein yield, how they are connected to the milk production, to the organic matter, to the crude protein, and the natural detergent. And if you see, it is basically from the extensive to the less extensive to the intensive and highly and very highly intensive kind of management that you have, and it depicts a positive correlation with the quality that is adjusted with the yield, including the yield of metabolizable energy, milk production, potential crude protein, presence of potential crude protein, organic matter, and even neutral detergent fiber, independent of the management, how intense the management is. So if we could make our management much better, and in that case, we could actually harvest much more from such a treasure trove that we have from the plant kingdom, not only in India, but worldwide. So now, little bit, how can we connect this plant diversity, and how does this plant diversity affect our revenue? So if we try to see this diversity, because of the number of species that we have, also is directly linked to the revenue, because the biomass is directly related to the economy of the economy. And in that case, you know, our economy started from the carbon era, and then we have different economy and bioeconomy now, and from the fossil economy that we are moving forward. And here also plant diversity has played major role and will play major role in the years to come. And the forest and also the agriculture, industrial energy, and that will be the key driver of the bioeconomy in next 10 years of the civilization that we would be seeing in next 10 years. And that possibly would be one very, very key contribution of the plant diversity towards attaining the sustainable development goal that we all know to be attained by the next 10 years, by 2030. And now let me try to say that we are losing the diversity. How do we conserve them? Basically, there are two ways that we conserve all the diversity. One is ex situ conservation, where you could see we have such nice gardens. I have just taken the major gardens that we talk about, but trust me, India has many more other gardens, but the major ones that we know is the Botanical Garden of Ooty. We also have J.C. Bose Indian Botanic Garden in Kolkata, where you have this very huge banyan tree, more than 200 years old, still alive and giving us the shade, below which many of us possibly with this pleasant shade that we all grow. Now we have Directorate of Wheat Research in Karnal, we have Lloyd Botanical Garden in Darjeeling, and then National Botanical Research Garden in the NBRI, Lucknow, also Central Rice Research Institute, Cuttack have a garden. When we talk about in situ conservation, India also has different national parks, biosphere reserves, and wildlife sanctuary, where we keep on trying to reserve our plant diversity, as a whole. Now, how do we do that? A small cartoon that how public and the gardeners do these things, the in situ conservation or the ex situ conservation, through the horticulture and molecular breeding, and through a public outreach program that has been categorized over here. Now, if you go to the next slide, what we then find is that not only that we need to do ex situ and in situ gardening, but also it's very, very important that we preserve our germplasm of the plant. Let me tell you, India has thousands of germplasm of many plants. Even if I go to those 12 plants, a handful of 12 plants, we have more than 1 lakh rice germplasm in our country, which are not only the cultivated variety, but the land races and many other wild relatives. And those wild relatives, those land races have never been tapped, and I don't think even in our lifetime, but maybe our next generation lifetime, those will be the treasure trove that we need to use our modern tools and technology to understand. And how do we utilize this diversity for us, for the planet Earth, for the plant themselves. And in that case, this slide actually I'm not going to read out, but tells you that what all those germplasm conservation that we have in different institutes of our country, that it has been done not only in our country, in the UK, in the US, in France, in Belgium, Japan, Canada, and every country. So everybody in the world is now always new, but this diversity loss is coming as a threat, and therefore we need to preserve them. So as you know that the poet, she said that Diana Ackerman said that we are losing biodiversity globally at an alarming rate, and we need a cornucopia of different plants and animals for the planet's health and our own. And then at the other hand, this person, Paul Watson, he says we are close to losing our essential diversity. Look at our wheat crops. It's very pertinent to a talk of plant diversity. We rely on a handful of grain crops and plants that we have refined and bred over hundreds of years, which is very true. The wheat we are having, the rice we are having, coming from the Green Revolution in 1950s, from the Norman Borlaug and the M.S. Swaminathan's work, we have now many different other mega variety of rice, 20 mega variety of rice, but still so much to tap. Very recently, Department of Biotechnology came up and gave 8 crop as a genetic germplasm characterization. Hopefully in 5 years, we would come up using many new sequencing technology, that what are the treasure we had in all those are in the germplasm there. Now, there are different threats. There are majorly, these are the threats. One is anthropogenic climate change, wind, many other things, pollution, invasive species, overexploitation, fragmentation, degradation of our lands, and habitat loss. These are basically 6 very important criteria that determine this kind of a threat to the plant diversity, out of which the maximum comes from the anthropogenic climate change and the minimum is due to the habitat loss. Now, if we try to see, then what do we do and where are we today? So let me tell you, we have a gold mine which is still unexplored. We have many things to do, and it's for all of you, the students and researchers who are listening this talk today. It's for you that you have a gold mine with you. Let me tell you this gold mine and how these gold mines have been categorized. And in that case, it has been told, and if this paper, if you read ecography paper by Cornwell, and they actually categorize this shortfall in our knowledge of the plant kingdom in different type of shortfall. The first type of shortfall that we know is linear shortfall, and that is the area outside the accepted taxonomic thing, that is the ash color highlight. What is this? This is the diversity for which we have no name. Even we have those plants, but we do not know their names. So they are the linear shortfall, because linears actually started the classification. We then have a Darwinian shortfall. And what is the Darwinian shortfall? And that is the area outside the genetic data footprint. And that means it is the yellow region. And that region is basically the diversity for which we have 0 genetic information in our gene bank. So then as we move on, it is a Wallacean shortfall that we call, and that is the area outside the geographic data footprint. And in that case, it is the outside the green area. That means it encompasses the little bit of the blue and the red and the rest of the area. And that is basically where we have 0 location information, that where are those plants. And therefore, it is a geographic data footprint that needs to be traced back. Now, then the 4th one is the Raunkian shortfall, and in that case, it is the area outside our trade data coverage. And that trade data coverage, as you could see over here, that is a blue vein, and in that case, outside that, it is there, which is where we do not have we have 0 record of functional trade data. Now, the last one is the Venn shortfall, and that is a culmination of this all first 4 shortfalls that I talk to you now, is the area outside where there are 4 kinds of knowledge interest, the area that overlap where species are considered broadly covered. So therefore, you know, it's a gold mine that we are sitting on, and why not tomorrow we start actually venturing them, to explore them, to understand more. And in that case, how do we do that? Then what should be the future focus? As Marie Curie said once, nothing in life is to be feared. It is only to be understood. Now, I think now it is the time to understand more so that we may fear less. And if I now try to put Marie Curie's word in the context of the plant diversity, I think that, you know, it is time to make use of all advanced tools like genomics, proteomics, metabolomics, system science, AI and ML to discover all those treasure troves and unknown species and catalog them to unearth their potential for civilization to continue on this very nice planet Earth where we all live, and in some other planets maybe in future. But then what should we ask the questions? The questions that we should ask is, have the tropics as a whole and each tropical region separately been mainly a sink or a source of plant diversity? Is high diversity correlated with high speciation and or low extinction? What is the molecular basis of this plant diversity? And as you could see at the left-hand side, you see the yellow zone, and these yellow zones are basically ceratophyllates, eudicots, rhozoids, and asteroids, and they have been actually studied enormously. However, as you move to the green part, you find monocots and comminates, and they have been studied, but not in such great detail. When we come to the blue zone, that is the magnolioids and chloranthals and many others, particularly algal species, we do not know possibly even 1% of those which both present in the land and the water. And therefore, many things are there in front of us to discover. How do we do that? So whatever I talk to you, if I try to now sum up, riding the waves, we have seen a formative years where it was all description of specimens and speciation from the 15th to 18th century. That was the beginning of the academic study of the plants. Also, we then moved to the transition period, I think, which is speciation to heredity to chromosome to genetics, and then the middle days of genes and genomes, and 19th to 20th century. And then right now we are in the present era where the genomics, epigenetics, many other omics, which are decoding not only the genome, but the proteome and also chromatin and also the metabolome. And in that case, what happens, there are many tools that have been used to assess this plant diversity, from the phenotypic analysis to the barcoding to the sequencing, now to the next generation techniques. These are the plants under each of the heading where these techniques have been used only to find out their diversity. So these techniques have been used at least in one plant for, say, Arabidopsis. Now, how do we utilize our AI and ML is that there are many things: breeding, speculation of forest thread, genetic diversity, high throughput phenotyping, all can be done using the AI and ML. And then if we could do all this, we could find out what we don't know today. In that case, the biodiversity can be the boon for the bioeconomy, and by the study starting from genome and connecting to the phenome, and then we have a very different species diversity and use it for crop improvement, understanding novel pathway, and by making models thereafter. So I would like to close by my talk by quoting three of the quotes. One, I took it from our Honorable Prime Minister and President of CSIR, Council of Scientific and Industrial Research, when actually he inaugurated the CSIR way back in 1950, 26 November, he said, how to combine that past with the present and the future, how to have changes and how to have continuity and how to have both together. So now, if I then go back and see what Albert Einstein said once, he said, we shall require a substantially new manner of thinking if mankind is to survive. That is what we need to do, of newer thinking to connect the old to the new. And I would end my saying by quoting Buddha and saying how beautiful is a plant. And he says, if we could see the miracle of a single flower, clearly our whole life would change. And that is where we are today. Plants are diverse, and plant gives us so much beauty to see and many things to unearth, which can connect us with everything on this planet Earth. They give us food, they give us oxygen, and they make us live on this beautiful planet Earth from their beautiful action. Thank you.
Thank you, Professor Chakravarti, for the very nice talk, very inspiring. And the importance of plants in the lives of anyone, in particular in human life, cannot be overemphasized, perhaps. And that's what we learned from you today. There is a question from Sangeeta. Ma'am, why is Scalacea called Darwin finches of the plant world?
Yeah, actually, we know about the Darwin. Charles Darwin made his Galapagos Island travel, and then he found out, and we only know the Darwin finches, but this is the plant that he actually found, and it is a perfect plant example of a differentiation into different species occupying different ecological niche, and he found that is present in the Galapagos Island, and that is why it is known as, Scalacea is known as the Darwin finches.
Okay, from Shabarish Kurup. Since scientists have proven that life forms exist on Mars, can we plant seeds in Mars?
That is what may be our expectation. Till not, people could do it. NASA, as I said, trying to do their space mission. We don't know when that have very different atmosphere. Now, as I said, this all diversity grows because of the ecological niche that these plants are there. Right now, I don't know whether these plants can go, possibly not, and there may be some other species that might be there. So we still don't know, and much more work, much more science need to be, discovered before we can plant anything in Mars. But maybe a day would come when Earth is all, there is no space in Earth. Possibly somebody will go to the Mars. Definitely not me, but somebody else in future generation.
Yeah, let's hope so. From Wakar Yunus, what's your opinion on fleece flower plant with its human-shaped fruits?
This I won't be able to tell just now. I have to see. I can, if you share your email and send me my email, which I have shown in the first slide, I'll definitely write you the answer. I don't want to answer something which I do not know exactly, but I'll be very happy to write an email to you.
Sangeeta SK asked about Scalesia already, but she has another question on Scalesia. Is it the first most kingdom found in that era?
No, we can't say, because in that era there are other things also. So it's not the first in that era, because he found it as an example. But in Galapagos Island, he found many other plants. So it is not only Scalesia.
From Madan Gopal, all food directly or indirectly come from plants. Since about 12 species of plants are only used for food by humans, why are other species not explored, as growing population needs more food instead of working on biotechnology and changing their traits?
What happens is that it is basically the domestication that makes that what all we are having now. What I said, that there are 4 million or maybe many more yet to be discovered, but then those are not yet domesticated. So based on our cultivation history, based our breeding history, we come and narrow down the base. I do agree that we might go and try to figure out, and that is what many of the countries are doing, and we are not the exception. In India also, we have started cultivating millets. Millets were there, jawar, bajra, but not that those were in Rajasthan, some places, but not in the whole country. But now we are growing them in many more to have, as maybe that can be used in place of rice and wheat. However, let me tell you one thing, that it is also our habit, right? So if suppose you are a rice eater, you are given a millet, whether it will take some time for the people to also adjust with their own food habit. I agree that we need to, increase the diversification of our food basket. Nevertheless, we also need to improve our existing, those 12 or 16 crop variety on which we actually survive today. So both are needed. Nothing is actually, nothing can be thrown away, and one above the other. Both are needed, and definitely there are many things, many species we can definitely look for their utilization. For example, cassava is a tuber crop which is being taken as a staple food in Africa, but here only in Kerala you see cassava, but still Kerala, all Keralite people have their staple as rice, not cassava. So it's somewhere the food habit also come into the picture.
There are some questions on YouTube, so let's take some of them and then we can come back here again.
Are hybrids or BT crops a threat to plant diversity?
From Manish.
Yes.
Yeah, so this is, I think, kind of a question that comes from years to years, but I don't think it is a threat to any diversity, because, the BT crop, if you see the way we were losing, there's cotton before the BT cotton came into the cultivation. So now we do not have that much threat. First thing, let me tell you one thing. Any improvement we do in to make a plant better, we always should remember anything we do, we cannot actually play with their, the yield. So if the yield is not there, no farmer is going to cultivate any of the crop species. So therefore, BT has been taken by the farmers very nicely because it gives protection the bollworm so nicely. But it is not a threat. Only thing that people might think that if we are only growing BT, are we losing the cotton diversity? Yes, sometimes I would say land space is basically limited. So maybe we would lose some of the diversity of the cotton, but then we have to see there is a balancing act. So what do we need now? More cotton which are in regular cultivation, regular need, and then as I have shown you, there are in situ conservation, there are ex situ conservation, there are germplasm repositories. We can still keep all the other variety of the cotton in those places so that we do not lose them. At the same time, we get the benefit of the BT cotton in the field for the farmers as well as for its use.
Yes, somebody asks whether you can throw some light on parasitic plants.
Abhishek Mishra.
Best thing, I think, Ascuta, and in some place I have shown, but now I don't remember which slide, some of the parasitic plants that I have shown. So you see there are parasitic plants which grows on another plant. And why they're parasite? Because they actually draw their nutrition from the plant, host plant on which they grow basically. But many times they also are beneficial. So best example I think is Cuscuta. So that much we can say, but there are many other literature, and if you want to read, you can go and read about them. It will be difficult to keep on telling in details.
There is a question from Shabarish Kurup. Do blue oranges exist?
Blue oranges. This also I need to check. I know blue tomatoes are there now. To my knowledge, I don't think we have blue oranges.
Maybe you can enlighten us on blue tomatoes, since none of us, I at least I have not seen any blue tomatoes.
It's actually not blue. It's basically it's anthocyanin pigmentation. The tomatoes were made from the UK group, John Innes Center, and they made actually more anthocyanin into it. It's purple color basically. So you have those tomatoes are already there where you have more of anthocyanin, so that if you have those tomatoes, it's nutritionally enriched. So the whole biochemical pathway of the anthocyanin pigmentation have been altered in those tomatoes by engineering a transcription factor. And that's a very nice product, that genetically engineered tomato that we have now.
There's a question from Piyush. Piyush says, can we increase crop productivity without harming biodiversity?
Absolutely. There is no question. Always over the years, we have actually increased our productivity. And, there was a famous quote by Malthus, who was an economist and who said that population grows in geometric progression. However, our crop production grows in arithmetic progression. And that is so true. And in that case, we need to actually make the different kind of diverse plants, and we improve plants either by molecular breeding or by genetic engineering, or today by CRISPR-Cas9 technology, by gene editing. But whatever we do, diversity will not be threatened, because diversity is untapped. Diversity is a treasure trove, a gold mine, which is all throughout there. But if you make this kind of improved variety, there will never be a threat to the diversity. Only thing, the question comes, we are using the limited land space for cultivating a handful of some species, and we are not cultivating the others. Now the question is, we need to cultivate them because we need to feed our population. So we need to have the productivity, and we are having productivity very nicely, and we need to still make more production because the population will increase. But we can always preserve this diversity in the gene banks, in the in situ conservation. Whenever those will be needed, we can take them out. However, before we do anything, it's very essential that we do basic study onto all those diverse crop species, which is, it would take centuries, actually, I would say. It's not a work to be done in a day or a month or a year. It is a day, it is a work for centuries.
Okay, Sarvesh Shukla. Ma'am, is it possible to cultivate all crops and plant diversity only through hydroponics techniques?
No, Sarvesh. Reason because hydroponic technique, you have to actually develop the technology. So we do not have that technology, hydroponic technology, for all the crop species and all the diversity. We possibly can do that, but it would require time. It would require to optimize the technology, because the hydroponic was used for few crops initially. Later on, even hydroponic has been used now to produce potato tubers, which is known as microtubers, that you can make potato tubers in hydroponic, and that is possible. But to do that, it took years to develop that technology, because every plant, if you now see, even when you do a tissue culture, every plant needs a different protocol, because every plant inside has different chemistry, as I told you, and therefore the hormones, which are the molecules which actually are intimately linked to the plant growth and development, are very different in their concentration, in their the ratio of maybe auxin and cytokinin. So in that case, yes, I won't say that you can't do that, but it would require a lot of work, a lot of finding out the protocols and establishing them to make hydroponic cultures for all the species on the earth.
Yeah, I think we will close with that. Thank you very much for the inspiring talk, and you have a great question answer session, because you have been asked questions for nearly more than 20 minutes now, and that I think is a record. So thank you very much for this talk.
Thank you.